Welcome to the pre-class video for dental biochemistry. I'm Professor Fedora Sutton, and over the next 20 minutes or so, I want to give you the chemistry you'll need before we meet for Lecture 1. You have seen videos like this before. This is about acids, bases, and buffers. There are different versions, but what I want to do here is different from what you've viewed online. I want to build it in a way that goes somewhere specific. As dentists in training, we're going to study it with respect to the mouth, towards enamel, towards saliva, towards the reason a local anesthetic sometimes fails. And I want to spend real time on something more acid-based. Lectures skip almost entirely the role that amino acids and proteins play as buffers. That is a very important matter, both in your blood and on the surface of a tooth. When you come to class, we will not reteach this. We will start from it, do the calculations, and take it into the oral cavity. So watch the whole thing and bring your questions. Here's the plan. Part 1. Why hydrogen ion concentration matters so much to the body and why we bother converting it into this thing called pH instead of just quoting a concentration. Part 2. The definitions. You need to know what is an acid, what is a base, what is a conjugate pair, and what really separates a strong acid from a weak one. The answer to the last question is not what most students think it is. Part 3. Buffers. What are they? What is the single most important rule about them? Which is that a buffer works best near its own pKa. We'll cover Part 4, the 3 buffer systems your body actually uses, and in Part 5, where I'll spend most of the time, we look at amino acids and proteins as buffers. This part connects directly to dentistry. Let's start with a comparison. If you drew your blood right now and measured sodium, you'd find about 142 millimoles per liter. Potassium, about 5 millimoles per liter. Both of those are perfectly ordinary numbers. However, when you measure a hydrogen ion, the value is 0.00004 millimoles per liter. There's roughly a 3.5 million times less hydrogen ion in your blood than sodium. But that concentration of hydrogen ions is very important. Your body will tolerate a fairly wide swing in sodium. It will tolerate a swing in potassium, though less than it would in sodium, but it defends hydrogen ion concentration ferociously within a range so narrow that if you move outside it, you become unwell within minutes. The obvious question, why? Why guard so jealously something there's so little of? So, H plus concentration is 4 times 10 to the minus 8 moles per liter. Since you don't want to work with such a number, it has been compressed. There are 4 steps. Step 1, the concentration itself, 4 times 10 to the minus 8 molar. Step 2, you take the log. The log of 4 times 10 to the minus 8 is minus 7.4. Notice where that comes from. The exponent minus 8 gives you most of the answer. The coefficient, the 4, contributes the remaining 0.6. So it is not simply counting zeros. The coefficient matters, and it matters by more than half a pH unit. As you do your calculations, remember this. Step 3. Since nobody likes to work with negative numbers on a chart, the minus sign is moved across. So the pH is defined in terms of negative log of the hydrogen ion, and that equals ion concentration, and that equals 7.4. Then the name is given. How is the name? The lower p means the negative log of, so pH means the negative log of hydrogen ion concentration, which pH equals 7.4. So pH is not a separate physical quantity. It is a concentration that has been compressed by an algorithm. Because we move the minus sign across, the scale runs backwards. A lower pH means a higher concentration of hydrogen ions. Look at the 3 panels. At pH 7.4, we have 4 times 10 to the minus 8. Drop to pH 6.4, and we have 4 times 10 to the minus 7. That is 10 times more hydrogen ion, not less. If it drops again to 5.4, and we're at 4 times 10 to the minus 6, 100 times more concentrated than where we started. Because this is logarithm, every whole pH unit is a factor of 10. So let's answer the question, why does the body guard hydrogen ion so fiercely when there is so little of it? The answer is proteins. Think about what a protein is. Your DNA is transcribed to RNA, which is translated into a chain of amino acids, and that chain does nothing useful as a chain. It has to fold, and the way it folds is determined by the chemistry of the side chains hanging off of it. Negatively charged side chains are drawn towards positively charged ones. Groups that dislike water bury themselves in the interior. Groups that like water sit on the surface. As a result, the three-dimensional structure is formed, and out of this structure come the functional parts, such as the active site of an enzyme, or the pocket where a hormone binds, or the pore of an ion channel. Whether a side chain carries a charge at all depends on the pH of the solution it's sitting in. Change the pH and you change which groups are protonated. Change which groups are protonated and you change the pattern of attraction and repulsion. Change that pattern and the protein folds into a different shape or fails to fold properly at all. And if it doesn't fold properly, it doesn't work. So a shift in blood pH isn't a chemical curiosity. It's a simultaneous structural attack on every enzyme, every receptor, every transporter in your body. That's what's at stake. That is why 7.35 to 7.45. So remember side chains of amino acids and the charges of the side groups. In part 5, it turns out to be the mechanism of buffering itself. Now the definition. You must know, and you already know, an acid is anything that donates a hydrogen ion. A base is anything that accepts one. That's the Bronsted-Lowry definition. It's the only one we need in this course. Notice what follows from that. If an acid donates its hydrogen ion, what's left behind is a species that could accept one back. So acids and bases always come in pairs. We call them conjugate pairs. HA on the left, H plus and A minus on the right, with a double arrow between them because it goes both ways. Three pairs you'll see repeatedly: carbonic acid and bicarbonate. That's a big one in blood, and we'll spend real time on it. Dihydrogen phosphate and monohydrogen phosphate. That's the phosphate system. Important inside the cells, in urine, and as it happens, in saliva. And the third one is the one to pay attention to: protonated histidine and neutral histidine. That's an amino acid side chain, and it's doing exactly the same job as the other two. Keep that third one in mind. We'll come back to it. Also note the difference between strong acids and weak acids. Strong acid dissociates essentially completely, but hydrochloric acid and water, and effectively every molecule gives up its proton. It doesn't come back. The pKa of hydrochloric acid is somewhere around -7, which is just a way of saying the equilibrium sits almost entirely on the right. A weak acid dissociates only partially. Some molecules give up a proton, some don't. And crucially, the reaction runs in both directions at once. It sits in equilibrium. Now, that reversibility is not a defect. It is the whole point. An acid that can both release and recapture a proton can respond to conditions. If the solution becomes too acidic, it takes protons back. If it becomes too alkaline, it lets more go. A strong acid can't do that. It's already given everything away. So every buffer in your body is built from a weak acid. That's not a coincidence. This is a requirement. You will sometimes see a weak acid defined as one whose pKa is close to physiological pH, somewhere around 6 to 8. That's not right. That statement describes what makes a good buffer at blood pH. It does not define weakness. Lactic acid, for instance, is the acid produced by the bacteria in dental plaque, the acid that dissolves enamel. The single most important acid in this entire course has a pKa of 3.86. It is a weak acid. It's nowhere near 7.4. Weak means incomplete dissociation. It does not mean mild, it does not mean dilute, and it certainly does not mean harmless. So what is a buffer? A buffer is a solution that resists a change in pH. And the way it does that is by containing both members of a conjugate pair at the same time. A weak acid and its conjugate base sitting together in the same solution. Think about why that combination works. If somebody adds acid, a flood of hydrogen ions, the base form is sitting right there to absorb them. If somebody adds alkaline, the acid form is sitting right there to donate hydrogen ions and make up the difference. You have a reserve in both directions. And that leads directly to the most important rule about buffers. And you have to be able to state this code. Look at the equation on the screen. pH equals pKa plus the log of base over acid. Imagine the two are present equal amounts. Base over acid equals 1. The log of 1 is 0. So pH equals pKa, which means when the pH of the solution equals the pKa of the buffer, the pair is exactly 50-50. Maximum reserve in both directions. That is the point of greatest buffering capacity. Move away from the pKa in either direction, and one of the two reserves starts running down. By about 1 pH unit away, you've lost most of your useful capacity. So the rule is simple. A buffer works best when its pKa is close to the pH you're trying to defend. This explains why one particular amino acid does almost all the work. So, your body runs 3 buffer systems. You need to know all 3. The bicarbonate system with a pKa of 6.1. This one operates in extracellular fluid, in your blood principally. The phosphate system, pKa 7.2. This one works inside cells, in the renal tubule, and in saliva. And the protein system, where the working group is the histidine side chain, which has a pKa around 6. This operates inside every cell in your body, and in the red blood cell, it operates on a very large scale. Now, look at those pKa values against blood pH of 7.4. Something should trouble you. We just established the rule, a buffer works best when its pKa is near the pH being defended. Phosphate at 7.2 is beautifully placed. Protein at around 6 is a fair distance away, and bicarbonate at 6.1 is more than a full pH unit away from 7.4. And yet bicarbonate is the most important buffer in your blood by a wide margin. It looks like a contradiction. It isn't. Here is how we resolve that. Here's the bicarbonate system written out in full. You need to know that from memory. Carbon dioxide plus water in equilibrium with carbonic acid, in equilibrium with hydrogen ion plus bicarbonate. 2 double arrows. Everything moves in both directions. The first step, carbon dioxide combining with water, is slow. Left to itself, it is far too slow to be of any use for moment-to-moment control of blood pH. What rescues it is an enzyme called carbonic anhydrase, which accelerates that step enormously. Without the enzyme, this system could not do the job it does. Now the resolution of the puzzle from before. Bicarbonate's pKa is 6.1. Blood is 7.4. By the rule we learned, that should make it a mediocre buffer. And in a sealed tube, it would be. But your body is not a sealed tube. Look at the left-hand end of that equation. Carbon dioxide is a gas. It is volatile, and you are breathing it out continuously right now. That means the acid side of this system is never allowed to build up. Whenever hydrogen ions push the equilibrium leftward and generate carbon dioxide, you exhale it, and the reaction keeps running. That is what's called an open system. Its power comes not from a favorable pKa, but from the fact that one of its components is under continuous, independent physiological control. So the answer is bicarbonate wins not because of its chemistry, but because of its plumbing. The elegance of the bicarbonate system is that 2 entirely different organs each hold one end of the same equation. On the left, the carbon dioxide end belongs to your lungs. If your blood becomes too acidic, you breathe harder. You blow off carbon dioxide. The equilibrium shifts leftward. Hydrogen ions are consumed and pH rises. If your blood becomes too alkaline, you breathe less, retain carbon dioxide, and pH falls. This is fast. You can meaningfully change your blood pH within a minute or two just by changing how you breathe. Nothing else in the body is that quick. On the right, the hydrogen ion and bicarbonate end belongs to your kidneys. When the blood is too acidic, the kidney excretes hydrogen ions into the urine and simultaneously reabsorbs and regenerates bicarbonate, restocking the reserve. This is slow, hours to days, but its ultimate capacity is much greater than the respiratory route because the kidney can go on doing it indefinitely. So respiratory, short-term, fast, limited. Renal, long-term, slow, powerful. One more caution. It's a favorite exam trap. What these two systems do is called compensation. Compensation moves pH back toward normal. It does not remove whatever caused the problem, and it usually does not return pH the whole way back to 7.4. If you ever see a compensated value sitting perfectly at 7.4, overshooting past it on the other side, something else is going on. Compensation mitigates, it does not correct. The phosphate buffer system. Dihydrogen phosphate on the left, in equilibrium with hydrogen ion plus monohydrogen phosphate. The pK is 7.2. Now 7.2 against a blood pH of 7.4 is almost perfect placement. This ought to be a superb buffer, except for one thing. There simply isn't very much phosphate in plasma. Good pKa, small quantity, so in blood, its contribution is modest. Where it comes into its own is inside cells, where phosphate is present at much higher concentration, and in the renal tubule, where it accepts hydrogen ions being excreted into the urine. There's a third place that matters to us specifically. Phosphate is a significant buffer in saliva, and it is most important at low salivary flow rates, when bicarbonate is at its lowest. That's overnight, when your mouth is driest and least protected. So phosphate, ideal pKa, limited by quantity in blood, but a serious player inside cells and in the mouth. Now we come to part 5, and this is the part I most want you to take away. Amino acids and proteins are buffers. And once you say it out loud, it's almost obvious. A buffer is a weak acid with an ionizable group, and amino acids are absolutely covered in ionizable groups. So the interesting question isn't whether they buffer, it's which groups actually do the work at the pH of your body. Let's go through them. Every amino acid has an alpha carboxyl group with a pKa around 2.3. Compare that to 7.4. It's more than 5 pH units away. At blood pH, that group is fully protonated, and it has essentially nothing left to give, effectively no buffering. Every amino acid also has an alpha amino group, pKa around 9.6. That's more than 2 units above 7.4, so it's fully protonated and holding on tight. Again, essentially no buffering where we need it. So free amino acids floating in solution are actually rather poor buffers at physiological pH. Now look at the third row. The histidine side chain, the imidazole ring, has a pKa of about 6. Out of all 20 amino acids, that is the only common side chain that titrates anywhere near physiological pH. It is essentially the body's designated buffering reserve. Cysteine at 8.4 contributes something, but it's mostly protonated at 7.4, so its contribution is modest. And here's the structural point that makes all of this matter. When amino acids join to form a protein, the alpha amino and alpha carboxyl groups get consumed, forming the peptide bonds. They're gone. Only the two ends of the chain retain them. So in a protein, essentially all of the buffering capacity lives in the side groups. Let's stay with histidine because it earns the attention. The side chain is an imidazole ring, and it has a pKa of about 6. On the screen, you can see it behaving exactly like every other buffer we've met. Protonated form on the left, releasing a hydrogen ion to give the neutral form on the right, with a double arrow between them. Why does 6.0 matter so much? Because it is close enough to 7.4 that at body pH you have meaningful amounts of both forms present at the same time, not a 50-50 split. We're 1.4 units away, so the neutral form predominates, but enough of both that the residue has reserves in both directions. It can absorb hydrogen ions when acid arrives, and it can release them when alkaline arrives. No other common amino acid side chain can do that at body pH. Aspartate and glutamate are down at 3.6 and 4.3, fully deprotonated. Lysine and arginine are up at 10.5 and 12.5, fully protonated. They're all locked. Histidine is the one that's free to move. A side chain, your R group, that can pick up and hand off protons at physiological pH is precisely what an enzyme needs to do acid-base catalysis, to grab a proton from one place in a reaction, move it to another. Dr. Sharif will take that up properly in the enzyme kinetics lecture. I mention it now so that when you meet it there, you recognize it as the same chemistry doing a second job. If you remember one pKa number from this whole video, make it histidine at 6.0. So if histidine is the buffering residue, where in the body do we find histidine in really large amounts? Hemoglobin. And hemoglobin turns out to be the largest protein buffer you own, for three reasons. The first is simply quantity. Hemoglobin is present inside your red blood cells at an extraordinary concentration, and each hemoglobin molecule carries a substantial number of histidine residues. Multiply those together and you get the dominant non-bicarbonate buffer in your entire bloodstream. The second is location. Where does carbon dioxide from your tissues get converted into bicarbonate? Inside the red blood cell. That's where the carbonic anhydrase is. And that conversion generates hydrogen ions. So you have a reaction producing acid inside a compartment. And sitting right there in the same compartment is a very large quantity of the one amino acid built to absorb it. The buffer is exactly where the acid is made. The third reason is the most elegant, and it's a piece of chemistry I want you to look forward to. Deoxygenated hemoglobin is a better proton acceptor than oxygenated hemoglobin. Think about when that matters. Out in a metabolizing tissue, hemoglobin releases its oxygen and in the same moment becomes better at picking up hydrogen ions, which is precisely the moment when that tissue is producing carbon dioxide and acid. The molecule hands over oxygen and accepts acid in the same breath, in the same place, at the same time. We'll come back to this in protein 3, where you'll meet it under the names Bohr effect and Haldane effect. When you get there, remember it started here as histidine doing what histidine does. And now let us bring this to the mouth. This is dental biochemistry. Three things. First, what are the salivary proteins? Saliva is not just water and bicarbonate. It contains proline-rich proteins, staterin, histatins, mucins, and every one of those is a chain of amino acids carrying ionizable side chains. They buffer, and exactly as with phosphate, they matter most when salivary flow is low and bicarbonate is scarce. Overnight, when your patient's mouth is at its most vulnerable, the protein buffers are doing a disproportionate share of the work. Second, histatins. Histidine-rich proteins. The name tells you the composition. You'll meet them as antimicrobial peptides active against candida. But look at what they're made of. They're packed with imidazole groups with pKa values right around 6, which is right in the range where plaque acid does its damage. A peptide that is simultaneously an antimicrobial and a buffer sitting in the fluid bathing your teeth. Third, and this one is genuinely clinically applied, the bacteria in dental plaque metabolizes arginine and urea producing ammonia. Ammonia is basic. It raises plaque pH. So amino acid metabolism by the oral flora is actively defending the tooth. That is the biochemical rationale behind arginine-containing toothpastes. It's a real strategy in caries management. So here's the message of part 5. In the blood, bicarbonate is king, but bicarbonate barely penetrates dental plaque. Down at the tooth surface, in the place where caries actually happens, it is amino acids, in proteins, in peptides, and as substrates for bacterial metabolism that do a great deal of the defending. That is why I have given you this before lecture 1. Let's put the classification of disturbances on the same equation because it makes the whole thing much easier to hold in your head. There are 4 named disturbances, and they come from 2 questions. Is the pH too low or too high, and which end of the equation caused it? If the problem starts at the carbon dioxide end, the lung end, we call it respiratory. If carbon dioxide is retained, as in hypoventilation or chronic obstructive pulmonary disease, it pushes the equilibrium to the right, generates hydrogen ions, and pH falls. That's respiratory acidosis. If carbon dioxide is blown off, as in hyperventilation, from anxiety or pain or altitude, the equilibrium is pulled leftward. Hydrogen ions are consumed and pH rises. This is respiratory alkalosis. If the problem starts at the other end, hydrogen ion and bicarbonate, we call it metabolic. Gaining hydrogen ions or losing bicarbonate gives metabolic acidosis. Think diabetic ketoacidosis, lactic acidosis, severe diarrheal, renal failure. Losing hydrogen ions or gaining bicarbonate gives metabolic alkalosis. Think prolonged vomiting. Notice that I never had to memorize 4 separate stories. I read all 4 off one equation by asking which end moved and in which direction. That's how I'd like you to hold it. And do keep hold of that point about compensation. Whichever system is not at fault will try to pull the pH back, but pulling back is not the same as fixing. Okay, so let's look at all 6 things. 1. pH is just a concentration that has been compressed by a logarithm. A lower pH means more hydrogen ion, and every whole unit is a factor of 10. 2. The reason your body defends pH so tightly is that proteins fold according to the charges on their side groups, and pH determines those charges. A pH shift is a structural attack on every protein you have simultaneously. 3. Weak means incompletely dissociated. It doesn't mean mild. Lactic acid, pKa 3.86, is a weak acid, but it dissolves enamel. 4. A buffer works best when the pH equals its pKa, because that's where the 2 forms are present in equal amounts and the reserves are equal in both directions. 5. Bicarbonate is the most important buffer in blood despite an unhelpful pKa. And the reason is that it's an open system. The lungs are continuously removing carbon dioxide. And 6, and I want this to stay with you, amino acids and proteins are buffers. They do it through their side chains or side groups, and histidine at pKa 6.0 does nearly all of the work. In hemoglobin, it buffers your blood. In histatins, it buffers your saliva. And in plaque, bacterial metabolism of arginine and urea raises pH directly. So that's the chemistry. Here's what we do with it when we meet. In lecture 1, we'll do the calculations, working out the pH of a strong acid, of a weak acid, of a partially neutralized solution, and working out the net charge on an amino acid at any pH you are given. You'll want to be able to do those under examination conditions without a calculator, and I'll show you how. We look at titration curves and the isoelectric point, and at a distinction students routinely get backwards, which is that buffering is strongest at the pKa and weakest at the pi. And then in the mouth, we look at the Stephan curve. the critical pH for enamel, why Staterin holds calcium and phosphate in solution, and a question you'll meet in clinic within your first year, why a local anesthetic sometimes fails in an abscessed tooth, and what to do about it. Bring the 6 summary points and bring your questions, particularly on part 5, because the amino acid material is the part that will pay you back the most across the rest of this course. Thank you for watching, and I'll see you in class.